This is a bit of biology with Mr. Rock. Today we're going to be talking about meiosis, which is also cell division, but with meiosis, it is cell division in order to create sex cells, which is either a sperm or an egg. The way that meiosis is different from mitosis is we do start with one cell at the beginning. So we start with a parent cell, and this parent cell has 46 chromosomes if we're talking about humans. And in meiosis, the cell, the first cell, the parent cell, undergoes two divisions. So it's going to divide once, and it's going to create two cells, and then it's going to divide again, creating 4 cells at the end. Other things about it are these cells at the end, the 4 cells at the end, are going to be genetically different. And the cells at the end are each going to have 23 chromosomes instead of 46. The reason why is because we're talking about the sperm and the egg here to make a human being when they come together. So your dad is going to make sperm that has 23 chromosomes. Your mom is going to have an egg that has 23 chromosomes, so that when they come together and fuse, that human being can have 46 chromosomes. So new definitions that we have to talk about is first, in the last video, I explained what somatic cells were in mitosis. Now I'm introducing a term called gamete cells. And gamete cells are just sex cells, so either a sperm or an egg, and these sex cells are haploid. What the term haploid means is it's a cell that only has one set of chromosomes. So haploid cells are going to have half the amount of regular cells for their chromosomes. So diploid cells in a human would have 46, and a haploid human cell would have 23. Homologous chromosomes, I want you to think of this like pairs of shoes. So humans would have 23 pairs of shoes, and they got their left side of shoe, they got a running shoe from mom and a running shoe from dad, and they come together to make your shoes. And you have 23 of these. So homologous chromosomes are just the different kinds of shoes you have. So you have running shoes, you got left running shoe from mom, left running shoe from dad, and you could keep on going on with this analogy. But it's the same chromosome, it's the same version of chromosome, it just might have different DNA on it. And then finally, a tetrad here at the bottom, it is when homologous chromosomes, so the shoes, join together. So this is a picture showing all the different chromosomes that you have. The reason why I want to pull this up is it explains the idea of homologous chromosomes and then also a tetrad. So homologous chromosomes, there are 23 homologous chromosomes here. So there's chromosome 3, chromosome 4, chromosome 5, and you got one from mom, and you got one from dad. So these chromosomes together, like the pairs of shoes, they come together, and when these shoes pair up, something really unique happens, and it's called crossing over. Crossing over is a process where homologous chromosomes swap their genetic information. And when I say swap their genetic information, they actually trade the legs of their chromosomes with one another. So they trade their chromosome, and if you look at the picture on the left here, all the way on the left, you have a blue chromosome and a red chromosome. This is a tetrad, so this whole process is a tetrad. It's 2 chromosomes together. They swap their legs, and then now at the end, you have a blue-red chromosome, and then you also have a red-blue chromosome. And this creates a lot of genetic variation. So it's a good thing. That makes every sperm unique. It makes every egg unique, because that means no chromosome is going to be the same. So one thing I want to point out, before we get started on the process of meiosis, is you're going to see prophase 1 and then prophase 2. The reason why you see the number there is because each stage is going to happen twice, because in meiosis, the cells divide twice, and that's a unique thing about it. So things that happen in prophase that were happening in mitosis. Number 1, spindles form. Number 2, nucleus disintegrates. Number 3, chromosomes appear. All of that stuff happened in mitosis as well. So this is very similar to prophase in mitosis. The unique thing here is crossing over occurs. So crossing over, like I just explained, the chromosomes, homologous chromosomes, are going to swap legs, which is going to lead to genetic variation. This step here is the biggest difference between meiosis and mitosis. So this step. Crossing over is really important, but this step really helps students to understand how meiosis is different than mitosis. So in metaphase 1, chromosomes line up in the middle as a tetrad. So they're lining up as a pair, and the spindles attach to the tetrad. This is kind of a tricky concept, so I actually took the time to draw out this big, long picture. What this picture is showing, on the left, you have 46 chromosomes in the middle of the cell, and they're going to get pulled apart. And that is mitosis, and there's 46 chromosomes lined up in the middle. On the right side is meiosis, and you have 23 chromosomes lining up with their pair. So lining up as shoes, like left and right foot coming together. And you're going to have 23 chromosomes in the middle, and when you pull the chromosomes away, you're actually going to be pulling away a full chromosome. So anaphase 1, what's unique about it is you're pulling away a full chromosome instead of a chromatid. In telophase 1, nucleus starts to reform, and the formation of 2 new cells begins. So what we have here, meiosis 1 summary. Cytokinesis happens, and it creates 2 haploid cells. Haploid means half the set of chromosomes. And this is going to be unique because meiosis is going to happen again to both of the new cells. The second point there is the DNA does not replicate, or it does not duplicate as it goes into the second round of meiosis. So in mitosis, it replicated at this point. I'm saying no replication is going to happen. The cells are going to divide again. So now you see prophase 2, that means we're in the second round of meiosis. You have now 2 cells. In prophase 2, there's no crossing over. So there's going to be no swapping of legs between the chromosomes. And the nucleus goes away, spindles reform. Some of these characteristics, like for metaphase, there's going to be common themes. So a common theme in metaphase is things line up in the middle. So with metaphase 2, now you have the chromosomes lining up in the middle. It's happening to 2 cells. And when you see anaphase, the full chromosome is going to get pulled apart. I'm sorry about that. So when you see anaphase 2, the full chromosome is getting pulled apart. This takes us to telophase, PMAT, prophase, metaphase, anaphase, and then finally telophase. You're going to see the formation of 4 new cells. The nucleus is going to start to reform, and this is going to be the last step in meiosis. At the top here it says meiosis 2 summary, but really this is the overall recap and review of what has happened in meiosis. So the first thing that I want to point out is 4 new cells are made. We started off with 1 parent cell, and that 1 parent cell went through 2 divisions. And at the end, we now have 4 new cells. All of these cells are genetically different. So every single cell we created is genetically different because of crossing over and because it only has 23 chromosomes. And that brings me to my last point. These new sex cells that you've made, either a sperm or an egg, are haploid. So they only have 1 set of a chromosome. That is the process of meiosis. This has been a bit of biology with Mr. Rock. I'm signing off.